Hello boys, welcome back to another session of video tutorials. This is an interesting session. We are continuing with the various experiments conducted on DNA. So this session is meant for class 12th biology students and this is meant for 19th August 2020. Our memory helps us in reflecting upon what Griffith has done, what MacLeod Avery has done, and in this session we are going to discuss about another great interesting experiment that is conducted by two greatest scientists, Hershey and Chase. So this is famous as Hershey and Chase experiment. This experiment is conducted in 1952 and they worked on a virus that infects bacteria. So all of you know by seeing the diagram only, it looks like a satellite. It has got a head, it has got a collar, and it has got a sheath, and it has got fibers, tail fibers. So this is a bacteriophage. Though the name is bacteriophage, all of you know bacteriophage is a virus and that infects bacteria. So do not think that bacteriophage is a bacteria. Bacteriophage is a virus. So during infection, the bacteriophage first attaches to bacterial cell wall. As I said, on the bacterial cell wall, there should be some receptor cells. And normally the receptor cells are proteins. So the receptor cells, being protein molecules, welcome the bacteriophage, as you are seeing here in A, picture A. So the moment bacteriophage sits on a bacteria and the proteins present on the membrane of the bacteria welcomes it, immediately the bacteriophage enters. So just like COVID-19. The moment it enters, now what happens? It starts spreading itself by increasing its numbers. How does that happen? As you are seeing in B, there is a way to enter inside through the receptors. And once it goes inside, by utilizing the energy and the resources that are available within the bacteria, bacteriophage multiplies. Fission happens, multiple fission. And thus, the inner body, inside the body of the bacteria, number of bacteriophages will be n number. And after some time, you are seeing in figure E, the bacteria cannot accommodate so many bacteriophages within its body. So it has to blast. Thus, bacteria is getting killed. So the virus called bacteriophage is killing the bacteria by blasting it off. So this is how the infection starts. So the viral genetic material became part of the bacterial genome. That's how it is doing. It manufactures more virus particles using host content, as I said. So Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. So they conducted a beautiful experiment. It's called Hershey and Chase experiment. What they did is blenders experiment. What did they have done? They grew some viruses on a medium having radioactive phosphorus, P32 and P34. Some others on medium having radioactive sulfur, S17. So viruses grown on radioactive phosphorus have radioactive DNA. Why radioactive DNA? DNA is made up of polynucleotidal chain, we said. And a nucleotide, if you recollect from your memory, a polynucleotide or any nucleotide consists of three things. That is sugar, phosphate, and nitrogenous bases. So phosphorus is part of the DNA. Hence the idea that Hershey Chase got was that if we can use, instead of normal phosphorus, radioactive phosphorus, it gives a glow and it's a heavy one. So easily we can trace that radioactive isotope. So that's how they incorporated radioactive phosphorus in the DNA, but not radioactive protein, because they wanted to know exactly whether it is protein that is causing the infection or DNA that is causing. Because phosphorus is present in DNA, but in protein it is not there, as all of you know. So viruses grown on radioactive sulfur have radioactive protein, not radioactive DNA. So now they have taken proteins, and all of us know that sulfur is part of the protein. So there instead of normal sulfur, they used radioactive sulfur. So once the protein penetrates inside, along with the protein, radioactive sulfur also penetrates inside the host cell. And then, being radioactive, it gives a glow, it's heavy in nature, we can easily trace the elements of sulfur in that. So they used radioactive sulfur in protein, radioactive phosphorus in DNA. So what they have done, they have blended that. So in the picture 1 you are seeing protein codes of bacteriophages are radioactively labeled. So protein codes will be having what we said, sulfur. That means isotope of sulfurs are used. And these bacteriophages in picture 2 you are seeing made it infect the bacterial cell. And in third you are seeing no radioactivity enters the cells. The protein content is outside only. It is not entering inside the cell. That means they have come to a conclusion, Hershey and Chase have come to a conclusion that it is not protein which is causing infection. Then they repeated this experiment with one more. What is that? DNA of phases is radioactively labeled. Now this time they left the proteins. The radioactive phosphorus is incorporated in the DNA because phosphorus is part of DNA. Now they allowed the bacteriophages to infect host cell, bacteria cell. And now interestingly, in third diagram you can see the radioactivity enters inside the cell. That means we can trace the elements of radioactive phosphate inside the host. That means it is not protein, it is DNA which is causing genetic material. So radioactive phages were allowed to infect E. coli bacteria, Escherichia coli. The phages transfer the genetic material to the bacteria. The viral coats were separated from the bacteria surface by blender. That's why it's called blender's experiment. The virus particles were separated from the bacteria by centrifuge machine. So what is that we have observed? The centrifuge machine is something like a mixie your mummy uses in the kitchen. So bacteria, observation first, bacteria infected with viruses that had radioactive DNA were radioactive. No radioactivity in the supernatant solution. Bacteria infected with viruses that had radioactive protein were not radioactive, but radioactivity found in the supernatant. What is the conclusion? Since we are able to see traces of DNA having that radioactive phosphorus and no protein is involved because sulfur is not there, radioactive sulfur is not there within the host, it goes without saying and it gives a convincing conclusion that DNA is the infecting agent that made the bacteria radioactive.